Biology chapter 19 theory video 8 separating and amplifying DNA. So this is done by using the polymerase chain reaction which is referred to as PCR. So this is used in all the gene technology related related activities. So even if you have a single molecule of DNA, you can actually amplify it. You can create unlimited quantities of that DNA length. So this is an automated process that amplifies selected regions of DNA using alternate stages of polynucleotide separation, that is denaturation of DNA and DNA synthesis catalyzed by DNA polymerase. Now in this DNA amplification module, this is done inside a PCR machine. So initially you have to put a sample of DNA, then part of that is going to be amplified. Then there should be two different short lengths of the, please remember all of these things need to be more than trying to understand, you have to remember these as kind of steps and stories. You don't have to actually understand everything in details, but you will have to know what it means and what it says and what the next step is going to be. So this is more or less a theory lesson where because these are really complex processes summarized into several sentences. So if you miss even a single sentence, the entire thing might be wrong. So please make sure that you remember everything. So there should be two different short lengths of single-stranded DNA, and that is going to be the primers for the DNA polymerase. Then there should be DNTPs, that is free deoxynucleotide triphosphate molecules, that would provide the energy to form the phosphodiester bonds. Then a buffer solution of pH 7 to 8 should be there, and a solution of thermostable or heat stable DNA polymerase. So when the PCR machine is switched on, it will have very small tubes to maintain the temperature, and they will have these tubes would have only about 0.05 cubic centimeters of volume, and so they will be working with very small amounts of material. In the first step, what happens is the DNA is denatured by boiling it up to 95 degrees Celsius. It will be heated up to 95 degrees Celsius. So that will denature it, break in the hydrogen bonds between base pairs, and it would separate the double-stranded DNA molecule into two strands. And the base would be expressed, or the base would be exposed rather. So this is the heating of the DNA would denature it, so that will actually present the next steps to be carried out. So here, as you can see, at the top of this diagram, you can see the DNA is heated briefly to denature the DNA, which separates the double helix, right? Now, the stage 2 is going to be annealing. The primers bind to the base sequence on either side of the length of DNA which is being amplified. So the primers would bind to the base sequence, as you can see. So here in number 2, primer DNA added after cooling, complementary base pairing occurs. So then that is going to happen. Then DNA primers are required because DNA polymerase cannot begin synthesizing DNA without an existing strand on which to build. Primers are often about 20 base pairs long and have base sequences complementary to base sequences either side of the part of the length of DNA that is to be copied. Part of the length that is to be copied. The primers have different base sequences. One binds to the upward strand and the other binds to the downward strand, as shown in the figure. Attaching the primer requires a temperature of about 60 degrees Celsius. So this is what happens here. So what you need to understand is, what you need to remember is, primer DNA will be added after cooling, then the complementary base pairing would occur. So the DNA polymerase will use the free dNTPs to synthesize complementary strands of DNA. And now it will, in the end, when the two lengths of DNA has been copied, it will form part of two DNA molecules. There would be two DNA molecules. So it will be heated up again, and the cycle will recommence. So the stage 3 is going to be extension. The enzyme DNA polymerase then uses the dNTPs, like I explained to you earlier, to build new strands of DNA against the export ones. The synthesis of new DNA strands using DNA polymerase, known as extension, requires a temperature of around 72 degrees Celsius. The DNA polymerase used for this process come from microorganisms that have evolved to live in hot environments. So after the first cycle, DNA has been copied, then the mixture is heated again to begin the second cycle. So the heating will separate two strands in each DNA molecule. Primers bind themselves to start off each strand of unpaired nucleotides, and DNA polymerase makes the complementary copies. After this second cycle, there would be 4 molecules of double-stranded DNA. So this can go on forever. This can go on forever because you can make more and more copies from the original tiny little amount of DNA molecules that you had. So a single DNA molecule can be used to produce any amount of copies in a few hours. So this PCR technology has made it possible for us to get enough DNA from a tiny sample. That is how even a single drop of blood left at a crime scene would be enough for us to identify, because we can copy this into large amounts, and then we can carry out whatever the test that needs to be done. So that is then this analyzing of DNA is carried out by gel electrophoresis. We'll be learning about that in the next part. We will be, but what you need to understand is the steps used in PCR mechanism. So this Taq polymerase was the first heat-stable DNA polymerase to be used in PCR. It was isolated from the thermophilic bacterium Thermus aquaticus, which was found in, again, the Yellowstone National Park in United States, which is actually it is one unique place. Yellowstone National Park is actually a supervolcano, which is actually overdue in erupting. So it can also erupt at any moment, and if it erupts, that would cause a mass extinction level event also. That's a different story, but it has lot of hot water springs which are boiling actually. That is 100-degree water is available in those springs. So there are these different kinds of microorganisms developed in these places who are adapted to those temperatures. Now this Taq polymerase is really valuable for PCR because it is not destroyed during the denaturation stage, so it doesn't have to be replaced in every cycle. And its optimum temperature will mean that the temperature for the extension stage does not have to be dropped below that of the annealing process. So we can, efficiency can be maximized. However, this is not the only DNA polymerase that is being used for this process. Write down a note. Separating and amplifying DNA, polymerase chain reaction, within brackets PCR. This mechanism is used in gene technology to amplify lengths of DNA from very small amount of DNA. Any number of copies can be made within very short period of time. This is an automated process that amplifies selected regions of DNA using alternate stages of polynucleotide separation, denaturation of DNA, and DNA synthesis catalyzed by DNA polymerase. To the PCR machine, following are added. Number 1, sample of DNA. Number 2, two different short lengths of single-stranded DNA to act as primer for the DNA polymerase. Number 3, free deoxynucleotide triphosphate molecules, DNTPs, providing energy for the formation of phosphodiester bonds. Next one, a buffer solution at pH 7 to 8. Next one, a solution of thermostable, that is heat stable DNA polymerase. When the PCR machine is switched on, the DNA copying process is carried out in 3 stages. Number 1, denaturation. By heating to 95 degrees, DNA is denatured, which breaks hydrogen bonds between base pairs and separates the double-stranded DNA into 2 strands. Number 2, annealing, stage 2 annealing. The primer will bind to the base sequence on either side of the length of DNA by forming hydrogen bonds. This allows DNA polymerase to synthesize DNA This process requires about 60 degrees Celsius. Stage 3, extension. The enzyme DNA polymerase would use the DNTPs to build new strands of DNA against the exposed one. This requires 72 degrees Celsius. When this cycle is over, second cycle would begin. And complementary copies would be produced. After the second cycle, there would be 4 molecules of double-stranded DNA. As a result, we would be able to produce any amount of DNA from a small amount of DNA. Now, for example, in the first 2 strands were there in the first occasion. In the first occasion, there were 2 strands. Then these 2 strands became 8. 2 strands were copied into 4 strands. So that means there were double, that is two double-stranded DNA molecules would be produced. Then those 2 will produce 4 double-stranded molecules. So you can see that it is getting doubled each time. 2 becomes 4, 4 becomes 1 becomes, 1 strand becomes 2, 2 becomes 4, 4 becomes 16. Like that it will multiply. I hope you understood. If you have any questions, please ask. I'm sorry, it is actually not 16. 1 becomes 2, 2 becomes, there is 2 double-stranded DNA molecules. 2 double-stranded, initially you have a single double-stranded DNA molecule that becomes 2 double-stranded DNA molecules. So these 2 double-stranded DNA molecule will become 4 double-stranded DNA molecules. That 4 will be, it will have, the 4 double-stranded molecules will have 8 single strands. So they will together produce 8 double-stranded molecules. So it is getting doubled each time. I'm sorry for the confusion. I hope you understood. If you have any questions, please ask. Please do question number 9 and 10.